So, thank you so much for coming, guys, especially those of you guys driving a distance to come here. So, I'm really excited to see you all here. So, in this kind of preface talk, I'm hoping to give you a different insight into the biology that we see around us every day, and then also to prepare you guys a little bit for my talk at 2 o'clock so that will make a little bit more sense to you guys. So those are my goals here. So I think if you're going around, walking around, or going to the zoo or the arboretum, you can really be struck with how diverse all of life is, right? We've got bacteria, birds, fish, flowers, redwoods, insects. It's amazing the shape and the sizes that life comes. You might think they couldn't get more different from each other, right? You know, you look at a dandelion and a tree, those are both even plants and they couldn't be more different. But as a cellular biologist, you kind of see a different side of this, which is how alike we all are. And so I was hoping to give you guys a little glimpse of that today so you can see that we really share, I think we share more in common than we don't share. So, I'm going to start off here talking about the basis of all life on this planet, which is cells. Bacteria, people, plants, it's all cell-based. Even viruses, which don't have their own cells, it's still cell-based life for them to replicate and to live. So everything is made of these little discrete units. Interestingly, this is an onion skin peel. It's a really easy place to see cells. You guys can see each cell has its little nucleus in there. And it was actually in plants that cells were first observed in the mid-1600s, and then the cellular theory of life was developed, which we later found to be true for all of us. So if you think of the cell as like a little factory, there's tons of stuff going on in there, all sorts of processes, and all of the workers and the machines in the factory, for the most part, are a type of molecule that we call proteins. And it's the proteins that are defining what type of a factory this is. So it's the type of proteins that you have in a muscle cell that are going to make it be a muscle cell. It's the type of proteins that you have in a skin cell that's going to make it a skin cell, or a leaf cell, or a bacterial cell. This is what's going to make you be a human, and is what's going to make you have your different parts of your humanness. And you guys, you might not know how many proteins that you're already familiar with from your everyday life. So I'm just going to talk about 6 of the most famous proteins that you guys have probably heard about in the news and on TV, just to begin to give you an idea of how many different things proteins can do. So the first one I'm showing here is hemoglobin. So this is the protein in our blood. You guys probably hear about this when you're donating blood or whatever. This is a protein. It's in your red blood cells. 4 proteins come together. That's why it kind of has that symmetric shape, because it's actually 4 of them together in a group. And they're able to capture the oxygen from the air in your lungs and carry it throughout your body to supply oxygen. So insulin, maybe the most famous protein in the world right now with the prevalence of diabetes. This is 6 insulin proteins that, again, form together. That's why it's got that symmetry appearance. And so insulin, as you guys probably know, it's kind of like a letter that you put in the mail. It's a signaling chemical in your body to tell your body how much sugar your cells should be taking up from your blood. Collagen, I thought you guys might have heard of in your skincare products. So collagen is a structural protein, so it's giving the elasticity and the structure to our skin. Another famous structural protein you might have heard of is keratin. On your shampoo bottles, they're always trying to say they're going to help your keratin. I'm not sure if that's true or not, but so keratin protein is the major constituent of hair, nails, and animals' claws, feathers. So again, it's a structural comp. So very different than hemoglobin, which is not a structural component. I thought you guys might have heard of BRCA1 in the news recently. So this is a protein that helps prevent your cells from proliferating and dividing too much. So if you have a defective version of this protein, you are much more likely to get breast cancer. So Angelina Jolie found out she has a defective BRCA1 protein. That was why she got the preventive mastectomy. So I thought you guys might have heard about that in the news recently. And amyloid beta, so during the course of Alzheimer's disease, for reasons that are still not 100% understood, this protein starts to clump together in the brain, and those clumps are likely one of the main causes of dementia in Alzheimer's disease. So that's an example of a protein kind of gone wrong, causing disease. But these are all proteins. There are about more than 25,000 different proteins in your body. I'm just showing you 6 that I thought you might have heard of, and to give you an idea of how much stuff these guys do. So if these define our cells and define our bodies as what we are, where do they come from? What's the source of these proteins? So that's what I want to talk about next. So our genome is the instruction manual for our cells, and it's our genome that defines what proteins get made. And the process by which our genome, which is made of DNA, is turned into these proteins is called the Central Dogma. So we'll zoom in on this in more detail. And it might sound like a kind of ominous central dogma. It really is just that important and thrilling of a concept. All of life on Earth works this way. This is really going to explain to you guys, in one way, how life works. So I'm going to start off with kind of an overview of it, and then we'll go into it in just a little bit more detail so you can see what an elegant system it is. So our genomes are made out of DNA. So it would be like your whole chromosome is one piece of DNA. So it's very long. There are thousands and thousands of genes just back to back in our DNA. It looks like, the molecule looks like a twisted ladder. You guys are probably familiar with seeing it on, Nova specials and that sort of thing. But when we need to make more of a certain protein, the gene that corresponds to that protein gets made into RNA. So RNA is a lot like DNA. They both sound the same, right? RNA, DNA. They're both nucleic acids. They're really similar types of molecule. It kind of looks like if you took the twisted ladder and just cut the ladder in half. So it's a really similar type of molecule. It's not as long because your messenger RNA is usually just for one gene. And the fact that it's called messenger RNA is really a great description of it because you're just making a little copy of just that one gene that you need to bring into the cytoplasm to make more. Kind of like when the waitress takes your order, puts that little tag up, like, okay, we need a hamburger on table 2. So it is like a messenger. And it's this RNA that then signals the cell to make the corresponding protein. And we'll talk about that more in a minute. But it's really worth noting that protein, that word does not sound like the word RNA, and it's not. They're not similar molecules at all. It's really different structure. You see, it doesn't look like a ladder at all. It still has a linear chain kind of layout, as you saw in the RNA. You can see it squiggling across the page. But that's where the similarities end. It's a really different type of chemical. So how is something so different containing the information you need to make protein? That's what we're going to talk about. So let's go back up to the top in more detail here. So we're going to make a little analogy here where your genome is the cookbook with thousands of recipes, all the recipes that we need to make all the proteins that make you. So DNA, as you guys have probably heard this on TV specials as well, has 4, I'm calling them like info bytes. If you think of like a computer programming, there's 0 and 1. In this case, we've got 4 info bytes to work with, A, T, G, and C. This, I'm going to show a little diagram over here. So it would be much longer, obviously. And there's the 2 strands of it, so it's kind of like the 2 rungs of the ladder. So this is one rung of the ladder, and then this one down here is the other rung of the ladder. And each of these little base pairings is one of the steps in the ladder that you would put your feet on. And each step is made up of one of the 4 bytes, and it's a pair, so 2 of them together. And so every time there's a T on one side of the ladder, the other side of the ladder's got an A. And every time there's a C, there's a G. So it's always together, A and T, G and C, always together in those pairings. So it's a redundant system. You only need the information from one strand to tell what the other strand would be. So, and there's a lot of reasons for keeping it redundant, but I would guess, so when you were running the computer systems at Loyola, what was the minimum number of copies that you would keep of the important files? Probably always 2, at least. And so we also have this kind of redundant system. So I'm showing in the middle here some bold bases. So that's just to represent, this is like our little fictional gene. This is much shorter than a real gene would be, but I had to fit it on a PowerPoint slide. And the little bases that make up a gene are exactly the same as the bases that are in between genes. I just put them in bold for us to see, but they would be exactly the same type of molecules. And let's say this little mini fictional gene is the gene that we want more. We want to make the protein that gene encodes? Well, we're going to make it into RNA. So the process of taking the gene that you need in DNA and turning it into our little messenger RNA is called transcription. And this is, again, actually a really good word, because if you think of a secretary doing transcription, you know, maybe she's copying the handwritten note into a typewritten note, but she's just copying it letter for letter. She's not really doing like any broad thinking processes. And that's the same thing. Since the DNA and RNA are so similar, you're basically just copying it letter for letter into RNA. And since our entire genome and the DNA lives in the cell nucleus, this is also happening at that site of the DNA in the nucleus. So this is how that happens. So this is our DNA from the last slide, but now we're splitting apart the two sides of the ladder just in the area of the gene that we need. And then other proteins in the cell start building the RNA. So you can see it's A with T. We have also 4 bases in RNA. Instead of a T, there's a U, but it's really quite similar. You can kind of think of it as the same thing. The chemical shape looks pretty similar. But so you just start building it base for base. You could see how that goes. If it's a A in the DNA, you know, you get a T, that sort of thing. So we build our whole RNA messenger molecule here. And there we go. So we now have our single-stranded messenger RNA. So in our cookbook analogy, this would kind of be like if you went into the library, picked out the meal you want to make, jot down the recipe, maybe on a little card that you're going to actually take into the kitchen with you to start cooking. So this is just like a little quickie copy of your recipe here. We've got those same 4 info bytes. But now we're going to have to really change the language we're using. Because before we got our 4 info bytes, we're all just copying letter for letter. Now we're going to protein. So as I said before, this is a fundamentally different type of molecule. So this is more like cooking. You're not just copying letter for letter. You're now actually in the kitchen starting to make yourself a meal. So this happens in the cell cytoplasm, and it's called translation, which again is a pretty good description. You're going from one language, your nucleic acid, to another protein. So you're really making a big transition here. So unlike RNA and DNA, we don't have 4 info bytes. There are 20 amino acids, and this is true for all of life on Earth. There's a few exotic species out there that have an extra, so they have 21 or 22. But these 20 are the core of all proteins. And you know, you guys might have heard of some of them if you've ever, you know, those bodybuilders that get those amino acid shakes. Sometimes they've heard of the specific amino acids, but anything that you eat contains all of these. Meat has more of them, but they're all there all the time. So this kind of brings up a mathematical situation, though. So we know that we're going to use the information from the RNA to say which of these amino acids we're going to string together in which order. So our RNA only has those 4 info bytes, and now we're going to have to communicate which of these 20 amino acids we're putting on there. So if you think of those 4 info bytes, it's kind of like base 4. So you know, our normal math system uses base 10. So we've got 10 digits, 0 through 9, and with 1 digit, we could specify 10 different things, you know, the 0 through 9 things. With 2 digits, we can do 100 things. You know, 0 through 99 are all the different things we can specify with 2 digits. Okay, well here we've got base 4. So with 1 digit, we can only specify 4 different things. With 2 digits, we can only specify 16 different things. That's not enough. We got to get up to 20. So we're going to need 3 digits in order to be able to specify 20 different things. So sure enough, that's exactly how it works. So we're back at, remember our blue RNA molecule? So in translation, what the cell is going to do is recognize each 3 nucleotides in a 3-letter word. You know, so you could think of it as a 3-digit number or a 3-letter word. We call them codons. It's a word that it comes up enough that we have our own word for it. So each little codon is read, and then there's a corresponding amino acid. So AUG is always going to code for methionine. And so it starts, so methionine is one of the amino acids. AAG is always going to code for lysine, so it puts methionine, then it adds on a lysine, and down the road, kind of like adding little train cars to make a full train. So this goes along, and this is now a chain of amino acids. That's a protein. So this is how the information from the RNA is used to build a protein. So proteins are are usually much longer than this. In general, they're 100 to 1,000 amino acids long, so I couldn't fit them on the slide. All right, so each of these amino acids have a different chemical shape, and those attract and repel each other in ways that make this protein fold up to have a 3D shape. And that 3D shape is going to dictate what it does for a living. Is it going to be able to carry oxygen? Is it going to be able to signal your cells to take up sugar? It's that 3D shape that's going to dictate it. And it's the sequence of amino acids that determines that 3D shape. So maybe a kind of reasonable analogy would be if you had magnets of all different shapes that were beads, and if you thread the magnets onto a string, the string would like glom up into a shape, and that would be dictated on how those magnets were shaped and attracted each other. So for those 6 proteins I showed you in the beginning, we had those beautiful ribbon structures showing how that chain of amino acids wraps around into all these different shapes. We don't know the shape for most proteins. You know that we know them for those 6 most famous proteins I showed you guys, but we know that they all make some 3D shape. And if researchers work on it a lot, it is possible now to find the 3D shape of most proteins, but it's a lot of work. So, okay, but so based on those codons that have the 3 digits of the base 4 numbering system, you can actually make 64, so it's 4 times 4 times 4, 64 different codes, but there's only 20 amino acids, so what do you do with all those extra codes? Turns out it's a redundant code, so for example, all 4 of these different codons all code for a proline, and so you can see most all of the amino acids have multiple different codons that code for them. Methionine only has 1, so it's kind of a mixed bag. Some have as many as 6. Arginine's got 6 of them. But I want to point out something really incredible about this. This is the code that I use to make proteins in my own body, and all you guys are using. It's the same code that the E. coli in my gut are using to build their proteins. This is phenomenal. There are, of course, a few exotic exceptions that don't use this exact code. Even those exceptions only differ by 4 out of 64 of these. This is incredible. We're all, this is because there's no reason it has to be this way. There's no reason that AUG has to encode methionine any more than 911 doesn't have to call emergency, right? You could imagine going back in a time machine and convincing them to make it be 811. You know what I mean? It's just the reason that we all share this same exact code is because we all share an ancestor. We're all related to each other. That's why we're all using this same code. It's not like a rule of physics or chemistry that it has to be this way. So this is not just like finding out that all computers run on 0s and 1s. This is like finding out we're all running on Windows 7. You can take the coding sequence from E. coli and usually make that same protein in one of my cells. You can usually take the coding sequence from one of my cells and put it in a plant and usually make a protein from it. All the regulatory sequences have kind of diverged over the 3.8 billion years since our common ancestor. But this core central dogma is the same. So we really have a tremendous amount in common with all life on this planet. And when people talk about life on other planets, not only would it not have to be DNA to RNA to protein in the first place, but if it was, I see no reason why AUG would have to be methionine. Why not AGG, you know? So it's because we all share this common origins. All right, so the most, within cellular life, the biggest distinction that you can make is between prokaryotes and eukaryotes. And I wanted to go over this because especially in my talk at 2 o'clock, I'm going to be saying eukaryotes all the time. You guys are going to be like, eukaryotes? So prokaryotes you can kind of think of as basically bacteria, would be not quite a synonym, but almost. So close enough for us to talk about today. So they're much smaller. They're essentially all single cellular life forms. They don't have a nucleus or other membrane-bound organelles. Eukaryotes, on the other hand, these are a lot of the visual large things that you guys think of when you walk around outside. So plants, animals, fungus, the organisms I like to call the organisms previously known as protists, just like Prince. But these can be single cellular or they can be multicellular, and they're probably most of the types of things that you would think about walking around outside. But everything that I've talked about up until now is the same between these two giant groups of organisms. From here on out, though, I'm going to talk about some things that apply more to eukaryotes. So the common ancestor of all life, and this is a big window, but probably goes back about 3.8 billion years ago, whereas when these two split from each other, and then all of the eukaryotes went on to make these different groups, was probably more like 1 to 2 billion years ago. That gives you a scale. The whole planet's only about 4 billion years old. That gives you any scale at all. It's hard to keep in your mind. So yeah, so from here I'm going to talk about mostly some things that I think will help you guys out at 2 o'clock so that that talk will make more sense. Do you guys want to stop and ask any questions now? We can do a lot more of that at the end as well. All right. So my lab works on a plant called Arabidopsis. So I wanted to explain that because that also probably does not come up in the news or TV very often anyway. So think of Arabidopsis as kind of like the mouse, right? So you guys know a lot of experiments are done on mice, not because we're really interested in mice. I mean, they're great little animals and everything, but we're studying mice because they're easy to grow in the lab, they're small, they reproduce quickly, and things that we learn about mice are often true for other organisms, all the way from humans to other animals, all sorts of things. Well, that's Arabidopsis as well. We're not really interested in Arabidopsis. We're interested in Arabidopsis because it's so easy to use in the lab and tells us about all these other organisms, plants in particular, but we also learn things about all sorts of life. So for example, I showed you earlier that all life is made of cells, and that was first found out in plants, and later we learned that's true for all of us. And that's just one of many examples that were found in plants, but turns out to be true for humans as well. And I think it's important to note here that if we didn't share a common ancestor, right? If you didn't think humans were related to mice at all, why would you ever test drugs on mice, right? It wouldn't have any meaning for humans. But we know we are very closely related to mice. We're even related to Arabidopsis. And so that's why the type of science that we do using a model organism works, because we are related. We do share these things in common. So some of the things that make Arabidopsis so easy to use in the lab is it's pretty small. Actually, a little pot of Arabidopsis is about the size of a mouse, but it doesn't run around, so you don't have to put it in a cage. So that's a bonus. It has a fully sequenced genome, and that's been true since about 2000. So it's been a fully sequenced genome from back before everything had a fully sequenced genome. One key thing that you're going to see in a few minutes is really important is that we can add genes into Arabidopsis. So they call that transformation. It has a reasonably quick generation time. It's about 2 months. I actually think it's about the same generation time as a mouse, which is kind of funny. Makes a lot of seed. That's handy. Unlike a mouse, Arabidopsis can self-pollinate itself. That's handy for the genetics. So there's just we've got a whole bag of reasons why this is easy to use in the lab. So one of the key sets of tools that we're using to figure out what genes do is a toolbox that we call genetics. I'm just going to explain one aspect of it here today, but I think it'll help with my talk. So in this little analogy, the cell is like a car factory. So it's making cars. And here we've got a fully functioning factory that's making these very nice cars. This is what a normal car looks like, so we're calling this the wild type phenotype car. Well, let's go look at our factory here. We got these great factory workers. Each one of them has their own job, like proteins, that they're doing to build these cars and to make a working factory. And maybe we're trying to figure out how these cars are made and what these different workers do. And we're thinking, this guy here. So this guy's Larry. And it would help us to understand what's going on here if we knew what does Larry do in the factory. So one way to find out what he does is to take him out of the factory. So with cells, we're going to do this by mutating the gene that encodes him. So sometimes you can literally remove the gene. In other organisms, it's easier just to disrupt the gene, so like the equivalent of like cutting off Larry's head. In any case, you're getting Larry out of the factory, so he can't do his job. Well, now what sort of cars are we getting? Now we're getting cars with no tires, we can say, without Larry. So this would be our mutant phenotype, would be no wheels. And so from this, we can infer that Larry's required for tires. Now, it could be that he puts the tires on the car, maybe he builds the tires, but it looks like Larry's job is somehow involved with getting the tires on these cars. I just wanted to mention that this idea of analogy using cars as cells as factories was made by Doug Kellogg, who's a professor. One little caveat I wanted to mention here is most eukaryotes, not quite all of them, but certainly us and the majority of plants have 2 copies of every gene. We get one from our mom and one from our dad. That's true for Arabidopsis as well. Usually its mom and its dad are the same plant, but in any case, we've got 2 copies of Larry. So if you want to remove Larry from the factory, you better get rid of both of them. Otherwise, the other Larry is just going to do the job. So if you are wild type for the Larry gene, you have 2 copies of Larry. If you are heterozygous, you have one that's mutated or messed up in some way or missing, and 1 normal copy of Larry. And if you're homozygous for the Larry mutation, Larry is gone. You've gotten rid of Larry on both chromosomes. So those will come up later. And I just wanted to make sure it was clear when we're using these words, heterozygous and homozygous, we're usually talking about just one gene. So even though we're just talking about Larry, you can assume the rest of the factory workers are all there. We're just talking about the one gene. Okay, so this is going to relate back to the car factory, but we're going to take a little dip in the ocean here to talk about one of my favorite proteins. It's a really cool tool that we use in the lab. So it's called green fluorescent protein, or GFP. This was originally found in jellyfish in the ocean, but as I mentioned before, you can take coding sequences from one organism and generally put them in another and make the same protein. So this was done here. So this jellyfish has a more complex multi-protein system that allows it to glow underwater. You guys have probably seen those deep underwater, really beautiful pictures. This is just one protein that's part of that larger system. When you have this one protein on its own, it is able to absorb blue light, so blue photons, and then spit them out again with slightly lower energy, which would make them be a green light. So this is kind of, I mean, a lot of people call it a glow-in-the-dark protein. It doesn't literally glow in the dark. That's a decent analogy, but it only glows when you shine blue light on it. So it's kind of like, you guys seen those, those velvet black light posters? They don't literally glow in the dark. They only glow when you shine purple light on them. It's the same thing for this. So if, but we've got microscopes that can do this. You just shine really bright blue light on this protein, and it spits back out green. So this is really cool, but you're probably wondering, like, how is this helpful? Oh, I did want to mention, the first protein like this was green, but it's now been modified to be red. So there's red fluorescent protein, cyan fluorescent protein, magenta. We've got the whole rainbow now. But this is how this is useful. We are going to cut and paste different genes together, and that process of cutting and pasting genes together uses a different toolbox that we generally call molecular biology. There's some kind of overlap between the terms for these toolboxes. But this is how it's going to work. So we've got our little fictional fun gene from previously in the talk. And now this is not the full-length GFP gene. Again, I made it shorter so we could put it on the slide. And if we put these 2 genes right next to each other, when they go to be translated, it's going to make one protein. So all we could do is take this piece of DNA, put it into our organism of interest. That process of putting a gene into an organism is called transformation. Another kind of ominous word, but gets it done. Any lab-designed piece of DNA like this, we call a transgene. And once you put this into an organism, they call it a transgenic organism. So you guys probably hear that on the news all the time with this big Monsanto thing going on with the transgenic soy. It means something like this has been put into that soy plant. Once we can get this DNA into the genome, or at least into the nucleus of our organism of interest, ideally it's going to be treated just like one of its own genes. And so it's going to get turned into protein. So in this case, we've got our folded up amino acid sequence for our dark green gene of interest, we'll say, and then a contiguous protein that is able to fold up and emit green light. So now it's kind of like we've got our protein of interest over here. Like, let's say this is the Larry protein. He's now being followed around by a green light so we can see where he is. So if we have a cell that's expressing this, we put it under the microscope, we can now see where our protein of interest is. So in this example, this is a mammalian cell, and this is just from the internet. Whatever protein this is making this kind of spider web network throughout the cell. So the cytoskeleton of the cell, so it's kind of, you think of it as like the rebar that holds up cells, has this spider web arrangement. So with this one experiment alone, you would now think like, geez, I think our protein of interest is involved in the cytoskeleton. So you could see how valuable of an experiment this is. Sometimes just being able to see where your protein is in the cell can give you a lot of information about what it's doing. So it's like, oh, whatever protein from the internet this is, when we add this GFP tag to it, we see it's all over the cytoskeleton. That's probably has something to do with its function. So it can be a really valuable experiment. So how can we combine this with genetics? So if you remember, we've got our factory that's got all the workers except for Larry. Larry's missing, and it's pumping out cars with no wheels. If we're able to transform this cell, add in a transgene of Larry, let's go one step further. Let's make it be a fusion gene. So you see now Larry's got one of those light stick necklaces around his neck. So if we add Larry, GFP Larry, back into the factory, what we would hope is that now if he's a functioning, working copy of Larry, he should be able to go back to putting the tires on the car, and we should see a normal phenotype come out of this. So we can learn two things from an experiment like this. We have just confirmed that indeed it was loss of Larry that was the true cause for having no wheels on the car, and not something else going on that we weren't aware of. You know, you could imagine there's some other worker in the background that is also knocked out that we don't know about. But if it were some other worker, when we add Larry back in, it wouldn't fix anything. The fact that it did fix the cars proves that, yeah, it was loss of Larry that was causing the no wheels on the car. And what we also showed is that putting a glow stick around Larry, he's still functional. We did fix the car, so he's working even with that GFP tag. You could imagine that there are cases where putting two proteins together could disrupt their function. But an experiment like this can show both of those things. And so this is called rescuing a mutant, which is it's a pretty good word, right? Because when you rescue something, you fix it. And we're kind of like fixing the cell by adding this in. So pretty intuitive word. OK, so I'm going to totally switch gears here and just do a few little quick things that are going to come up at 2 o'clock. So you guys will know what I'm talking about at least a little bit at that time. So earlier when I was showing you the gene and you had all the little green bases and there were just those bold ones in the middle. Well, in eukaryotes, often the gene is split up by these little commercial breaks in the middle. So here in bold, we have bases that we want to encode protein with later. Then there's like a break. These are bases we do not want to turn into protein. And then more bases that we're going to turn into protein again. So we've got to get this guy in the middle out of here before we actually turn it into a protein. So this initial process of turning it into RNA happens the same as I showed you earlier. And now we've got this RNA that's got this break in the middle. It's at this level of where it's a pre-messenger RNA where we're going to remove the commercial breaks. So we've got these two pieces that we do want in the protein, and we're going to put those together, and this guy in the middle is going to get removed. So the pieces that get removed are called introns. The pieces that remain are called exons. And this process is called splicing, which is another good name for it. Because when they recorded movies on film, and if you had an outtake in the middle of the film, you would cut both sides of the outtake and put them back together, and you call that splicing out, that scene. It's a great word. It's the same sort of thing. So we're splicing out. This does not happen spontaneously. This process of cutting out this intron and very carefully and precisely gluing those two exons back together, because if you mess that up, you're going to mess up the whole protein. This is done by this huge complex. It contains over 300 proteins, at least in animal cells, almost certainly in plant cells as well, called the spliceosome. And here's another little cartoon kind of depicting the same thing. So in this cartoon, the exons are shown as boxes, but as we know, it's the same type of nucleotides as that make up all the rest of the RNA and the intron as well. And these little cluster of proteins here are to represent this giant spliceosome. And these are think of it like a group of factory workers that come together and saw out that intron and put the two pieces back together. So this protein complex is called the spliceosome. And spoiler alert, we think Captain might be involved in that, is why that's going to come up at 2 o'clock. So, okay, and the last thing I want to mention about this is one part of my talk at 2 o'clock is going to show something called a yeast-2 hybrid assay. It's just using a yeast cell is a little living test tube to look for protein-protein interaction between two proteins of interest from your organism. So don't be thrown off by the fact that it says yeast. I'm still looking at Arabidopsis proteins when I do a yeast-2 hybrid assay. And the way that this system winds up working, if your two Arabidopsis proteins that you put into the yeast cell, if those two proteins bind each other, the yeast cells can grow better on selective media. If those two proteins that you put in there don't bind each other, they don't grow very well on the special selective media. And one way that you can demonstrate this is you can, so yeast grow on the surface of agar plates. They do that on CSI, I think, too. And you've got your different yeast strains that you've put your pairwise proteins of interest into each of these different yeasts. And then you can mix them with liquid and put 1 drop of the liquid on the little agar plate. And then you do a 10-fold dilution and do the next drop. 10-fold dilution in the next drop, 10-fold dilution like this. So the reason to do the same yeast strain with these different dilutions is it helps you compare the growth better. So for example, if we compare these two down here, so this is the same strain just in decreasing dilutions, same strain, decreasing dilutions. If we only compared these first two spots, you'd be like, I don't know, it seems like they're growing the same. But when we go out to these more dilute ones, you can see the bottom one's growing much better. So that's the only reason we do these dilutions. So all you guys really need to think about for that is the more that they're growing on the special selectable media, the more that particular two proteins that are in that strain are binding each other. Does that kind of make sense? So yeah, so I'm hoping that you guys will hopefully be able to make sense of most of the talk at 2 o'clock now. There'll probably be some things, but just go with it. And I hope that you guys are kind of inspired about how much we all have in common with each other as well. So that's that.